Next speaker is Roberto Montoro, Ferrer from University of Amsterdam, again a graduate student from Dr. Kemp's laboratory, and he's going to talk about ALD organoids.
Well, thank you for your attention, for coming up today. I'm a PhD student at Stefan Kamp and Mark Engelen group at the team ALD. And today I'm going to talk a bit how we're trying to model adrenoleukodystrophy in vitro with cells. The previous speakers have already done a great presentation on ALD, so I'm just going to skip through this. A bit louder, okay. I'm going to skip through the ALD presentation because my previous speakers already did a good job. But just going to say that ALD has a major impact at the brain, spinal cord, and adrenal glands. All patients present mutations ABCD1, and there is no clear genotype and phenotype correlation. And ABCD1 encodes for the ALD protein, which imports very long-chain fatty acids into peroxisomes, and whenever mutated, they cannot get imported to be degraded, and then they accumulate in plasma and tissues. So many different models have been developed to study ALD, and recently we wrote a review on discussing how we've been trying to do this, but the main message is just that each of the models recapitulate a certain aspect of the disease, and we also saw a gap on iPSC models and a potential for them to study ALD. And although we got a lot of insights from these different animal models and cellular models, there are certain questions that still remain unresolved. The main one is why do some people develop one versus other form of the disease? What is the actual role of these very long-chain fatty acids in the pathogenesis, and how its accumulation then leads to the neuronal damage and the myelin loss. And that is where my research aim comes in, which is to model ALD in vitro with induced pluripotent stem cells, which I will come into a bit to explain what they are, brain cells, to then understand better the role of these very long-chain fatty acids in ALD pathogenesis. So what are these induced pluripotent stem cells? Here you have a figure where at the top of a hill you see what we can interpret as a cell, and during development this cell has different choices to make. This is a pluripotent cell, and depends on the choices that it makes, it can go to the different sides of the hill, and those represent different cell types. So these undifferentiated cell can differentiate to different cell types. For example, this could be a skin cell. And so previously we thought that this would be just one direction. However, the work of these two scientists, Sina Yamanaka and Gordon, they got the Nobel Prize because they found out that if you would that you could actually reprogram these differentiated cells to pluripotent state, that they could then differentiate again to all the cell types, and they got the Nobel Prize for this. So we're using this technology to study ALD, and the way we do this is we have donor and patient cells, in this case skin cells or so-called fibroblasts, and with these fibroblasts then we reprogram, so we get again the pluripotent cell in vitro, and once reprogrammed, once they are pluripotent again, then we can differentiate them into different either cell types in 2D, which are relevant for ALD. This could be brain cells, or even also in 3D, and I will come to these models later on, but they are so-called organoids. And I want to start with the 2D work that we've been doing. In this case, today I'm going to present the work on astrocytes. So a brief introduction on astrocytes in the brain and in ALD. So astrocytes are the major glia cell in the brain, and glia is contrary to neurons in this case, and glia stands for glue from the Greek, because at the beginning it was just thought to be the cells that would glue the brain together. However, decades of work have shown that they have very relevant roles in the brain, such as the regulation of neuronal communication, energy homeostasis, also involved in the blood-brain barrier and in myelination processes. And relevant for ALD, post-mortem studies show that astrocytes could have an early pathogenesis in the brain. So they had high stress markers in an area that was not yet demyelinated. This proneness to study astrocytes with our pluripotent stem cells. So what we did, we have 3 healthy donors and 4 ALD patients. And after reprogramming the skin fibroblast, then we make these astrocytes, these cell types. And at the bottom you see 3 pictures, and those are immunostainings, fluorescent immunostainings. And we can label these cell types with specific markers for the cells. In this case, GFAP, SOX9 and CD44, ID3 and nestin, to identify that we've actually done this differentiation. So once we had these brain cells, then we started to ask questions to them. And the first one is, in healthy astrocytes, is ABCD1 present to start with? And the staining here in the green dots are the ABCD1 labeled peroxisomes. And if we zoom in all the cells, basically they do present ABCD1. And once we knew that the ABCD1 is present, then the next question with an ALD background is, okay, if we have now an ALD astrocyte, does it also recapitulate the biochemical deficiency, typical of ALD? And in the graph in the middle, what I've plotted here is the C26-lyso-PC, the biomarker used in clinic. And you can see that whenever we have the genetic background of ALD with astrocytes, they have an accumulation of C26-lyso-PC. And this second graph on the right, what you have is 2 time points of the same, either like group controls or different ALD lines. And the first time point is day 97, differentiation in vitro days, and the second time point is 114. And what you can see is that in all the lines, more or less pronounced increase. The very long chain fatty acids, they don't get degraded, and they also accumulate through time. And we also wanted to, so since we have the fibroblasts, then we wonder how do they match up with the skin cells. And here what you can see is that the fibroblasts, regardless of the patient background, in this case 4 lines, they also have very similar very long chain fatty acid levels, C26 in this case, lyso-PC. However, when we look at the astrocytes, we see a variable increase of this marker. And this gives a solid ground to study these specific brain cells, because maybe some mechanisms are happening in brain cells that are not happening in fibroblasts. And then the next question will be, okay, so we know that there is this biochemical deficiency, and that the different genetic backgrounds within ALD have different C26 lyso-PC levels, but how do the lipid profile looks like? So we did lipidomics on these astrocytes, and we see a very clear elevation here in red. This cloud here on the right is lipid, all lipid classes and lipid species that are incorporated, have incorporated very long chain fatty acids. Interestingly, some of them are, for example, these ceramides, which are highly present in brain. And then the next question, the next logical question is, now that we know that these deficient ABCD1 astrocytes that we have accumulate the very long chain fatty acids, and they have the disrupted lipid profile, how do they support neurons? Because in the brain, of course, you have both cell types together. So the common experiment is to have a co-culture in which we have both cell types together, the astrocytes and the neurons, and the setup is to mix and match the phenotypes, so that we have, for example, control astrocytes with ALD neurons, and so forth. So this was all in 2D, in a monolayer of cells. But what we can also do is these induced pluripotent stem cells, we can also grow them in 3D, so in suspension. And in this case, I included a picture here. This is just the cell culture plate, and the dots that you see, the white dots in each well, that is actually developing organoids. And an organoid is a cell quality model that recapitulates certain parts of development of the tissue that it models, in this case, the cortex and the spinal cord. And today I'm going to focus on the cortical organoids and a brief introduction on corticogenesis. In this figure, you can see here at the beginning of development, these are the so-called neuronal progenitors or radioglial cells. They divide symmetrically to amplify the progenitor pool, and at some point during development, then they start to divide asymmetrically to make neurons. This process then is exacerbated during development. Other progenitors also appear, like the intermediate progenitors here, and also the so-called autoradioglia, another main neuronal progenitor in the brain, that then they all develop into the cortical plate. Now what I'm going to show you is pictures of the organoids that we have sliced and stained through different markers to study what are the processes going on within them. And here the first one that you can see is in red. Here is the so-called ventricle zone, which is mainly populated by progenitor cells, and in this case this is labeled by SOX2. And these progenitor cells, they produce the neurons that here are labeled by MAP2, which is a neuronal marker, and they differentially locate within these organoids. There are also surrounding ventricles, ventricle-like structure surrounded by a lumen, similar to what the ventricle in brain looks like. If we zoom in into this ventricle-like structure, you can also appreciate here, labeled by GFAP, that the processes of these main neuronal progenitors are also recapitulated, with the cell bodies being close to the ventricles, but then they have this extension of the bodies where the neurons also then can migrate to the cortical plate. And indeed, if you keep these cells longer in culture, what you get here labeled is the deep layer neuron, in this case by CTEP2, and also an intermediate progenitor by TBR2, which are again a bit spatially separated. And if you keep them longer in culture, you even get the upper layer neurons. So this is a bit of a recapitulation of the cortical layering of a brain. It's not as perfect as a six-layer brain, 6 layers of a brain that humans have, but at least we have upper and deep layer neurons. And if you keep them longer in culture, you also get the glia cells. So in this case, the astrocytes here on the right, and the oligodendrocytes here by labeled by MVP. And after seeing that they recapitulated this, we also wonder if we keep them longer, how mature do they get? And for these, we also look at again the astrocytes, and here in GFAP you can see the morphology of these cells, which is greatly expanded, and they have I've isolated the staining here on the right, used with black and white for better visualization, and you can see that they have really long processes contacting a lot of probably also neurons here in red that are in their surroundings. We also did electron microscopy on these organoids to see at the very close level how they look like, and mainly what we see is that there seems that there are some synapse structures that you can see. For example, here this pre-synaptic ready-to-release vesicle pool, and we also observe some myelin structures here by the darker circles surrounding probably axons. And once we have this, then we started making questions again. And the first one is where in the... So here we have a clump of cells, right, that have different cell types, neurons, astrocytes, oligodendrocytes. So where ABCD1 is expressed. And so far we've been able to confirm this in the astrocytes. I don't know if this is going to play, but I'm going to try. It's not playing. Well, that's okay. But in the astrocyte it was very clear to see that the ABCD1 was also present in their somas. The neurons in the oligodendrocytes are a bit more difficult, but we're trying also to test if it's present or not. But this also say to us that ABCC1 is present in these brain organoids, and we run a pilot test in which we have lipidomics from these ALD organoids at the early stages. So this is day 25 and day 50, and what you mainly have here is progenitors and neurons. Day 25 mainly progenitors, and then at day 50 you also have some neurons. And we start to see some mild, in this case, lipidomic changes, but this correlates in the sense that this is a developing structure that recapitulates early development, and with all these, after checking all these things, then it prompted us to then go for the big study, which is the one that I'm currently doing. So I'm just going to tell you how it looks like. For this study, we've included 3 healthy donors and 10 ALD patient lines with different phenotypes, so we can then run statistics and check for differences. From these iPSCs, I'm currently making cortical and spinal organoids, and the idea is to collect at different time points samples to run lipidomics, so we can study a lipid profile as well as the transcriptomics, so the RNA profile within all the assays. And currently it's around day 130, so I hope that next year I can present this data also here. And just to give you an idea how large for this study, we need to collect actually around 2,000 organoids, but it's on the way. And with this, I'd like to thank the lab, thank the ALD group, and also Stem Cell Lab, where I mainly do my work. And thank you all for your attention. And if you have any questions, I'm happy to answer.
Thank you. That was great. Questions? Any online questions? And I just noted you said controls. You talked about isogenic controls or regular controls?
No isogenic control, no.
Yeah. So that's one thing.
Healthy base, healthy donor, yeah.
And I saw on a couple of your slides you had stain for MVP. So, so are there oligos there, or are there myelin-producing oligos there yet, or not? I guess it's very early still, 60 days.
Yeah, totally. So with the electron microscopy pictures here on the right, what you can see is that we have a darker staining, and these resemble myelin sheets, so compacted sheets of membrane, and these within are indeed the myelinating oligodendrocytes. This is at day 160 differentiation days, and we're going to take them until day 200. Hopefully that there is even more myelination by then. Yeah.
I have one more question, and so does this do these include microglia at all?
Not yet. But there's a new project with another PhD in the group that is actually going to look into it. Yeah.
Okay. Yeah, we played around with that a couple of years ago. Maybe good to just check protocols as well. Yeah. Okay.
I'll ask you a question.
Sure.
So how long do you keep them going? Do they live for like 7 days, or and how much time does that give you in terms of doing these experiments for the mechanisms in these mini brains?
Can you repeat it for the online people?
Yeah, so the question is how long you can keep the mini brains or organoids in culture, and in this time window, how can you study them, I suppose. Yeah, not 7 days at all, no. So for example, this one that I've been showing is 160 days, and the plan is to take them to 200 days. So it is indeed a very long protocol that you need to be patient to. But that is also the cool thing with it, because then we're gonna have longitudinal, more or less data, in which we collect the organoids, at different time points, and study how, for example, the lipid profile changes on time.
Yeah. Christina?
I mentioned briefly spinal cord organoids. Can you comment on what those look like, the organization, and how those grow?
Yeah, so the cortical ones are very well defined because they also have this lumen and everything. In the spinal ones, they also do develop a lumen, but the protocol that I am currently doing is not specific for either dorsal or ventral spinal cord. So we have a mix. We have a mix of, again, the brain cells, so neurons and oligodendrocytes and astrocytes. But for example, neurons, here you can find motor neurons, for example.
Any more questions? Oh.
Here, I'll ask one more. That's really exciting, and I hope it will get to the point where we can recapitulate some disease features in the system. But I'm wondering whether, like in the mouse, where the phenotype is probably more subtle than it is in humans, whether you could do anything more provocative to challenge the system. For example, knocking out ABCD2 and then comparing cells that have or don't have ABCD1, or changing the nutrients in the media to somehow provoke more dependence on elongases and elevated VLCFAs. Do you have any thoughts on that?
Yeah, it is indeed a possibility, but I think we're first going to characterize how it looks like just with the genetic background of ALD, and afterwards then think indeed if we need to induce a bit more stress to these cells to study pathogenesis. But first we need to check what pathogenesis is going on.
Of course.
Yeah.
Thank you.
It's already a very stressful job to grow these things. I mean, it's very painstaking work to grow these organoids for hundreds of days and feeding them. So it's nice work. Thank you.
Good. Yeah.